Introduction to chemotherapeutic drugs. Chemotherapeutic drugs are medications used to treat infectious diseases, cancer, and other conditions by interfering with the growth and replication of harmful cells, microorganisms, or pathogens. The term, chemotherapy, traditionally refers to cancer treatment but has since expanded to include treatments for bacterial infections, viral infections, fungal infections, and parasitic diseases. These drugs act by targeting specific cellular processes in pathogens or abnormal cells, leading to their death or inhibition of growth. Chemotherapeutic drugs can be broadly classified into several categories, depending on their mechanism of action, target, and the type of organisms or cells they are designed to treat. These include antibiotics for bacterial infections, antivirals for viral infections, antifungals for fungal infections, anti-cancer agents for cancer treatment, antiparasitic agents for parasitic infections. Mechanism of action of chemotherapeutic drugs. Chemotherapeutic drugs are designed to target and disrupt specific biochemical pathways or structures within cells or microorganisms. Common mechanisms of action include 1. Inhibition of cell wall synthesis, e.g., penicillins and cephalosporins for bacterial infections. 2. Inhibition of protein synthesis, e.g., aminoglycosides and macrolides for bacterial infections. 3. Disruption of nucleic acid synthesis, e.g., fluoroquinolones and antimetabolites. 4. Inhibition of cell division, e.g., taxanes and antimetabolites for cancer treatment. 5. Inhibition of specific enzymes, e.g., protease inhibitors for HIV. Introduction to protein synthesis inhibitors. Protein synthesis inhibitors are a class of chemotherapeutic drugs that specifically target and inhibit the process by which cells build proteins. These drugs can be used to treat bacterial infections or certain cancers, as they disrupt the ability of harmful cells, like bacteria or cancer cells, to produce the proteins necessary for survival and replication. Protein synthesis occurs in two main steps: transcription, the synthesis of messenger RNA, and translation, the assembly of amino acids into proteins based on the mRNA template. Protein synthesis inhibitors typically target various steps in the translation process, which is mediated by the ribosome. Protein synthesis inhibitors can be classified based on the type of ribosomal subunit they affect, bacterial or eukaryotic, and their specific mechanism of action. The main classes of protein synthesis inhibitors are. In this next video we can watch the bacterial or eukaryotic protein synthesis. Thanks for joining us. Like, share, and subscribe the Professor A. Sick for more amazing science videos.